Good morning, everybody. My name is Tovit Rosenzweig. I'm from the Ariel University in Israel. First, I would like to thank the organizers for the opportunity to present our study. And today I will share some of our data about Sarcopoterium spinosum and its potential use for the treatment of glucose intolerance, insulin resistance, and fatty liver. Sarcopoterium spinosum, also known as thorny or spiny barnet, is growing in the Mediterranean area, in the wild area, and is used for many years by Bedouin traditional medicinal practitioners for the treatment of diabetes. However, very few scientific evidence was documented over the years, and you can see here two interesting documentation with very dramatic titles: a sensational drug, new agent against diabetes, and Poterium spinosum, a potential cure for diabetes. These documentation are from the 60s and 70s of the previous century, and actually very few scientific evidence is included to validate the anti-diabetic properties of the plant. This more or less was the situation when we started our studies on Sarcopoterium spinosum. Before going deep inside the data, I would show the current regulatory status of this plant. You can see what is the situation in the European Union. The root bark of this herb is listed on the Belfrit list of botanicals. We are using this part of the plant, the root bark. And in the United States, presently there is no regulatory provisions identified for this plant, and there is no record of product of this plant being sold ever in the United States. During the year, we published most of our data. Today, I will present some of this, and of course, we have many additional data, but I will not present anything that was not published or is not covered by a patent. The outline of the talk will be, first I will convince you about the need to develop new drug for the treatment of glucose intolerance, insulin resistance, and fatty liver. Then I will show you the data, proof of concept, and then I will answer the question why to develop this plant towards the regulatory path of botanical drug. So, what is the need? I'm sure that everybody here in the audience is aware of the pandemic of diabetes all over the Western world. There are hundreds of millions of people suffering from this disease, and the predictions are that the numbers will be increased further. We know that type 2 diabetes is accompanied by several very severe complications. So this is very severe. We also know that there are several anti-diabetic drugs available for treatment approved by FDA and other agencies. So if there are drugs, what is the need? We can look on this graph. It was published 5 years ago. I don't have yet more recent data, but to the best of my knowledge, no dramatic change was made during the last years. What we can see here that around 40% of diabetic patients included in this survey adequately monitored or regulate their blood glucose. This was measured by hemoglobin A1C levels, which were lower than 7%. On the other side, 60% have higher hemoglobin A1C level, indicating that they have poor regulation of their blood glucose despite using anti-diabetic drugs. So this data emphasize the need to develop additional anti-diabetic drugs, and specifically it is important to develop new drugs with a different mechanism of action. So I hope that shortly I convinced you that still, despite availability of anti-diabetic drugs, still there is a need to develop additional strategies for treatment. And now I will focus on the proof of concept for the bioactivity of Sarcopoterium spinosum. I would like to give a short background about the disease. Type 2 diabetes is developed in high-risk individuals suffering from metabolic syndrome. This syndrome is a risk factor for the development of several additional morbidities, not only type 2 diabetes, including cardiovascular disease, stroke, certain types of cancer, and non-alcoholic steatohepatitis, and several other neurological disorders. In order to treat metabolic syndrome, it is important to understand the underlying pathologies that contribute to the development of this syndrome. These factors include major 5, having at least three of these 5 factors, including low HDL cholesterol, high triglycerides, insulin resistance, hypertension, and obesity, visceral obesity. If you have 3 of these 5, this is enough to diagnose the presence of metabolic syndrome. In addition to these 5 parameters, there are additional contributing factors to the metabolic syndrome listed here, including prothrombotic state, oxidative stress, inflammation. But as I mentioned, insulin resistance is one of these factors, and it's a major one. And we believe that if you want to give a good treatment to the type 2 diabetes and other comorbidities of the metabolic syndrome, you better target the underlying pathology, mainly insulin resistance. Today I will show you our data about the beneficial effect of Sarcopoterium spinosum extract for the treatment of insulin resistance, for the reduction of blood glucose, also amelioration of inflammation and oxidative stress. And I will show you that accordingly we found improvement in type 2 diabetes and in non-alcoholic steatohepatitis. All data was done in preclinical studies in animal models and in vitro models. So we used 2 different mice models for the study of insulin sensitivity and glucose tolerance. The first model was a genetic one. These mice developed type 2 diabetic as a result of genetic disturbance. You can see here glucose levels of mice. The top curve indicates glucose tolerance data of controlled diabetic mice. They have high fasting glucose, and after glucose load given, their blood glucose is increasing, and there is impairment in the ability of the cells to take glucose inside. So blood glucose remain high during the period of measurement. You can see here below the curve of metformin-treated mice. Metformin is one of the most used anti-diabetic drug, and we can see an improvement in blood glucose. But better than that is the data of mice given Sarcopoterium spinosum extract. We can see a significant reduction in their blood glucose, both fasting glucose and glucose disposal after glucose load. We then measure insulin sensitivity. This was done by insulin injection and measurement of blood glucose. We know that insulin is responsible for the uptake of glucose by cells. If there is impaired sensitivity, the ability of cells to take glucose is reduced, and this is exactly what we can see here. The upper curve is results of insulin tolerance test of diabetic mice. They have very low sensitivity to the hormone in compared to Sarcopoterium spinosum treated mice, which have very nice glucose response to insulin injection. You can see here the reduced level of blood glucose in these mice. In addition, and in accordance, we can see here a reduction in serum insulin in our treated mice. This is a good phenotype because we know that in subjects suffering from insulin resistance, there is a compensatory response of the pancreas to induce higher secretion of insulin, leading to hyperinsulinemia. And here we can see a reduction in blood insulin, indicating a better responsiveness to insulin. So there is no need to compensatory response. And this was in line with the lower area of pancreatic beta cells in pancreas in Sarcopoterium spinosum treated mice. The next model we used was the high-fat diet-fed mice. These mice develop obesity as a result of this obesogenic diet. And accompanied to this obesity, glucose intolerance is developed and insulin resistance as well. We can see here body weight is increasing in high-fat diet-fed mice. Body weight accumulation is much lower in mice given Sarcopoterium spinosum extract in addition to this obesogenic diet. More importantly, we can see here a reduction in their blood glucose, as was also demonstrated before in the previous model. We can see here normal glucose curve in lean mice given standard diet, and impaired glucose tolerance in mice given high-fat diet. When given Sarcopoterium spinosum in two different doses, we can see here an improvement in glucose tolerance. Again, this was accompanied by a correction of hyperinsulinemia. You can see here severe hyperinsulinemia developed in obese mice, but it was corrected when Sarcopoterium spinosum was given. So we show improvement in insulin resistance, improvement in blood glucose, lower severity of type 2 diabetes. And now I will show you additional effect of this extract, and this is on inflammation. We know that metabolic syndrome is promoted by chronic subacute inflammation. This is also called metabolic inflammation, and it's developed especially in adipose tissue of obese individuals. So it was very easy to measure the effect of Sarcopoterium spinosum on this metabolic inflammation. We isolate adipose tissue of mice, and you can see here in the bottom, you can see here adipocytes of standard diet fed mice, and enlarge the adipocytes as a result of high-fat diet feeding. You can see here that each adipocyte is bigger. But it's not only the hypertrophy of adipocytes that is observed. You can also see very important feature of this adipose tissue, there is between adipocytes there is infiltration of immune cells. This is exactly the inflammation that is developed in the tissue. When looking at adipose tissue of high-fat diet fed with the addition of SSE, our extract, still we can see enlargement of adipocytes. There is no difference between high-fat diet with and without SSE in their diameter, but there is completely no inflammation. This was quantified, both diameter of adipocytes and the presence of inflammation, and we can see here that although diameter is increased, there is no inflammation in Sarcopoterium spinosum treated mice. We also investigate the effect of our extract directly on immune cells in isolated macrophages. We measured certain biomarkers of inflammation, such as NO secretion, NF-kappa B activation, and specific pro-inflammatory cytokines. We induce inflammation in these cells, in these macrophages, by exposure to lipopolysaccharide, which is pro-inflammatory agent, and by with the addition of our extract, we can see prevention of this inflammatory response. So again, we can see anti-inflammatory effect of this extract. On the other hand, by measuring anti-inflammatory markers such as arginase 1 or IL-10, we can see here that our extract increases the expression of these anti-inflammatory markers. So we can also confirm the positive or beneficial effect of our extract on inflammation. Now we will focus on NASH. NASH is a non-alcoholic steatohepatitis. This is the hepatic manifestation of the metabolic syndrome. Actually, it is estimated that around 80% of diabetic patients suffer from fatty liver. A fraction of these individuals with fatty liver will have an advanced liver disease, which is NASH, and the problem is that NASH might be deteriorate further and further and might lead either to the development of hepatic carcinoma or hepatic failure. This is presented here, and we investigate the effect of our extract on the development of this fatty liver disease at different stages of severities. So first we can see here the effect on the basic stage of fatty liver. It's only steatosis. You can see here nicely histology of the liver. Here you can see normal liver of lean standard diet fed mice. And here you can see here the liver of high-fat diet fed mice. It is full with lipids. This is the steatosis of the liver. With the addition of our extract to the high-fat diet, you can see below that all doses given, all improved, not only improved, but completely corrected the histology of the liver and prevent the steatosis in liver. So we can conclude that Sarcopoterium spinosum can prevent the development or treat fatty liver. We next go to the next step of the disease, and we measure the effect of our extract on NASH. NASH is characterized not only by the development of steatosis, but also by hepatic inflammation. For that, we use different model. In this mice were given special diet named Western diet, and also fructose was added to the diet, and this is known to develop the non-alcoholic steatohepatitis phenotype. And we can see here the disorder in the liver in these mice. You can see here steatosis, and also these purple dots indicate the infiltration of immune cells towards the liver. So this is the phenotype of steatohepatitis, and when given increasing doses of the extract, we can see a gradual improvement in the histology of the liver. This was quantified by independent pathologists who grade the severity of the disease, and we can see here the improvement. And also we use biomarker of the presence of NASH. We measure the level of ALT and hepatic enzyme in serum, and it is known that ALT level is increasing with NASH, but we can see here reduction of to the normal of this enzyme by SSE treatment. This was also supported by molecular analysis of several genes that are involved in lipid metabolism in the liver. I will not go deep inside the data, but you can see here a set of genes that their expression was impaired. For example, here was impaired PPAR alpha by Western diet, and addition of the extract normalized the expression of most genes detected. Also, it prevents the increased expression of pro-inflammatory genes, for example, CD68, F480. We can see here an increase in their expression as a result of induction of NASH. And these are biomarkers of immune infiltration, and this was prevented by SSE treatment. And lastly, we can see here also normalization of the expression of antioxidant enzymes in the liver. We know that oxidative stress is developed in NASH, and we can see here alteration in expression of several antioxidant genes. Some are reduced, some are increased, but all were normalized by SSE treatment. So we can summarize the results so far. We found that spinosum improved insulin sensitivity, leading to better glucose tolerance. We found that SSE has an immunomodulatory effect and reduced adipose tissue inflammation, and it reversed hepatic steatosis and inhibited the progression toward NASH. Today we know that it is not enough just to say that a specific plant or a specific drug does something. It is also important to understand the mechanism of action. So we perform a lot of experiments to clarify the mechanism of action of the extract, and of course I will show you just a few of them. One of the important results we got was achieved by unbiased strategy of phosphoproteomics. We measure the phosphorylation of around 5,000 peptides, and using this was done in adipocytes given SSE treatment, and then we used a bioinformatic tool to predict which pathways are activated or inhibited by our extract in these adipocytes. And actually this is hypothesis-free approach, and we were very happy and very unsurprised to find that the top pathway predicted by the software was insulin receptor signaling, and the other signaling suggested later on were additional signaling that are associated with insulin signaling, indicating that our extract mimic insulin action in adipocytes. This was confirmed, and also we can see here by Western blot analysis that insulin phosphorylates its receptor. It's a biomarker for its activation. Also, there is a phosphorylation of another protein, namely AKT, which is downstream of the receptor. So this is insulin action, and you can see here a very similar picture obtained by SSE treatment. Again, supporting our hypothesis that our extract mimic insulin action and activate insulin. We measure glucose transport to adipocytes because this is one of the important physiological outcomes of the activation of insulin signaling. We can see here the effect of insulin for the induction of glucose transport, and SSE does the same, not exactly the same amount of effect, but very nice induction of glucose transport. And what is also very important, we can see here that there is an additive effect of SSE with insulin. So giving insulin and SSE together lead an extra effect. By inhibition of the receptor using a specific agent, we inhibit the phosphorylation of the activation of the receptor, and we can see here that this inhibition leads to inhibition of the downstream signaling. There is lower phosphorylation of AKT induced by insulin. And again, we can see here that by inhibition of the receptor, we also abrogated the effect of SSE on AKT, namely SSE also is using insulin receptor for the activation of the signaling. So we assume that insulin receptor is supposed to be the target for SSE action. So I hope that I convinced you about the need. I hope that by sharing you the highlights of our data, I convinced you that Sarcopoterium spinosum has a huge potential to be used as insulin sensitizers or insulin mimicking agent for the treatment of glucose intolerance and fatty liver. But now I just finish in the couple next minutes about the question, why botanical drug? Why not to isolate the active component and to sell this information to a big pharmaceutical company, and that's it. Why to go towards the regulatory pathway of botanical drug? So it's very simple. The answer is very simple. We know that botanical drugs are plant-derived, complex mixtures which may have synergistic effects beyond and be more affordable than their purified analogs. I will show you very few examples of that because this data was not published yet. And if taking the quote from the FDA definition, botanical drug products often have unique features, for example, complex mixtures, lack of distinct active ingredients, and substantial prior human use. Our product exactly fits to this definition. So during the last years, we made a lot of efforts for the isolation and identification of active constituents. Again, this was not published yet, and it's also not covered by patents, so I will share with you the detail. Just about the process, we are using bio-guided fractionation strategy. By fractionation, we are having several fractions. Their bioactivity is measured. We can see here that we have got very nice activity in certain fractions. These fractions were fractionated further, and again we measured bioactivity. We can see here again very high activity in certain fractions. This was again fractionated, but now you can see the problem. There is bioactivity, high bioactivity in fraction 6 and 7, but also there is some activity in additional fraction like fraction 9 or 3 and 4. So there is like a diffusion of the activity into more and more fractions. Also, when we measured it by a glucose uptake assay, we can see here that first fractionation leads to high bioactivity, but when doing additional fractionation step and subfraction 1, 2, and so on, we can see here again that when using high doses, there is bioactivity, but when using the actual relevant doses, there is reduced bioactivity, and actually we are losing the bioactivity when we are fractionating it into the into distinct chemicals. So we already identified one active molecules, and we also in the way for the identification of some additional active constituents, but as I show you, the results so far indicate that the activity is achieved by a concerted action of several compounds together. So this is it. Now, this is the situation where we are located now. We have a lot of additional data, and I can share in a different time. So it's a long road, but it's worth it. It's very interesting. It's very high promising. I would like to thank my partners from my student from Ariel University, my partner from Tel Aviv University, who contribute a lot for the chemistry part. I would like to thank financial support from State of Israel, and of course, investment and collaborations are warmly welcomed. And you have here the contact details. I can give additional information and continue our discussion later on. So thank you again for this opportunity.
Great. Tovit, that's a great presentation. I have a question. So is this product coming from a traditional herbal medicine, or you just discovered by your research team?
No, it was mentioned in several ethnopharmacological surveys done in Israel, in Jordan, in the Mediterranean area. So it's well known in the traditional medicine. And actually, in purpose, we decided to do all processing of the extract, at least at these stages, according to traditional medicine, because we didn't want to lose some products or some components that are required. I really believe in this, some kind of evolution of the traditional medicine. So we have to validate it and to use it smartly.
I see. And any other question? I have one more. So, you know, US FDA, they don't require mechanism of action or identification of active ingredient, although it's encouraged. If you do those, that will be helpful, but it doesn't really require to do that. Just, you know, just for you to know.
Yeah, what I understand that it's important to have a methods for standardization, and you know, to know that from season to season, from plant to plant, you have the same activity. So although you don't need a single or several specific active compounds for yourself, you need something to be used as biomarkers for activity.
Yes, I agree. Yes. So quality control is a very challenging issue for the herbal medicine. Yeah. That's all my question. Any other question from anyone?
I would like to comment. That was an excellent presentation. You and your team have done great work. the diseases that you are impacting, diabetes, fatty liver, those are areas that also here in America, the African American community experiences health disparities with those disease indications. And one angle that we are taking from a funding perspective is to align ourselves with institutions that are sourcing funding that impact minority health disparities. I've just recently signed a deal last week with a major pharmacology college here in the United States. And what we're going to do is do clinical trials of botanical drugs that impact the minority communities. And so it would be very interesting. What I really like also, I like everything about your approach. I represent a scientific research institute that specializes in absolutely what you're doing, capturing unique fractions of the extract and understanding what the molecular composition is of that extract so that you can more defensively not only articulate the science, but also to more defensively standardize your product during production. And I applaud that direction. We support that. And lastly, I would like to say that I like the fact that you've demonstrated that you've got a product that can work with insulin, because to be able to, to situate a clinical trial where we're able to show that you are able to achieve an additional efficacy, along with the standard of care is also very important here. So I look forward to working with you to see if we can advance these interclinical trials here for FDA approval. I like everything about your approach. The fact that you know more than what is required is better, more so than not. So we look forward to working with you.
Thank you very much. And you have my contact details, and we can continue discussion privately later.
Thank you. Mm-hmm.
Great. Thank you, Steve. Other questions? Okay. If not, thank you, Tovit, and for the presentation. We'll be in touch for the follow-ups. And thank you so much for your time.
Thank you.
Excellent presentation.